Hello, everybody. It's my great pleasure to introduce a very recent study working on the GABA and glutamate neurons in the ventral tegmental area regulate sleep and wakefulness. So why we work on VTA? We found VTA actually by accident. We started a long hypothesis-driven screening for wake-promoting circuitries in the brain, and we found VTA. So very brief introduction about the VTA. So VTA in the middle brain contains subregions, including paranigra, parabrachial, and intervesicular, and the rostrolinear area. And the different types of neurons locate differently in the VTA. So first, we profile genetically encoded VGLU2 or VGAT neurons in the VTA, and we verified that VGLU2 neurons were largely distinct from dopamine neurons. Only about 26% of the VGLU2 neurons express tyrosine hydroxylase, whereas VGAT neurons were largely distinct from dopamine neurons. Nearly none of the VGAT neurons express tyrosine hydroxylase. And VTA is has been intensively investigated its regulation for reward, aversive, and social behaviors. For example, VTA dopamine neurons receive excitatory inputs from lateral dorsal, tegmental, dorsal-raphe, and send projections to prefrontal cortex, nuclear accumbens, respectively, to regulate aversion or reward while dopamine signaling. And also, band nucleus can regulate reward or aversion through VTA. In addition, VGLU2 neurons in the VTA can regulate aversion via lateral or nuclear accumbens circuitries. But those behaviors require an arousal or wakefulness. And indeed, VTA dopamine neurons are REM and wake active. Optogenetic or chemogenetic activation of VTA dopamine neurons produce extended wakefulness while nuclear accumbens. So here, we ask how about the glutamate or GABA neurons in the VTA? Can they regulate vigilance states, and through what kind of specific neuronal circuitries? So to investigate this, we conducted chemogenetic experiments by delivering an excitatory jet receptor, HM3 DQ, into the VTA of VGLU2 Cre or VGAT Cre mice to target glutamatergic and GABAergic neurons, respectively. So this shows the VGLU2 mCherry expression in the intervesicular and paranigra of the VTA. This shows the VGAT neurons distributed averagely in the VTA. And then we gave CNO to activate VGLU2 or VGAT neurons in the VTA, and we found that chemogenetic activation of VGLU2 neurons in the VTA produced extended wakefulness for about 9 hours. However, chemogenetic activation of GABA neurons in the VTA induced long-lasting long-REM sleep for about 7 hours. So next, to determine the dynamic activities of those VGLU2 or VGAT neurons in the VTA, we expressed a calcium indicator by delivering a GCAM6 into the VTA, and we measured a fluorescent signal with EEG, EMG. As for the VGLU2 neurons, we found the fluorescent signal increased during REM sleep and wakefulness, which suggests the VGLU2 neurons in the VTA are wake and REM active. So this shows the summary data. But surprisingly, we found VGAT neurons in the VTA are also wake and REM active. So I add one more slide because we have more than 6. So, and then we did chemogenetic behavior mapping. So we inject HM3 into the VTA of VGLU2 animal, and then we gave CNO to excite the VGLU2 neurons in the VTA, and we mapped the C-force expression, and we found C-force increased in the lateral hypothalamus and the nuclear accumbens. And then we stained those C4 cells in the LH with orexin, and we found most of those C4 cells are orexin or hypercreatine positive. And to further identify the brain areas that participate in generating this VTA VGlu2 neuron-mediated wakefulness, so we performed optogenetic-based channelrhodopsin-based circuitry mapping by injecting channelrhodopsin into the VTA. And we found there was a dense fiber in the LH and nuclear combines. And to further test whether VTA to nuclear combines or LH circuitry could promote wakefulness, so we performed optogenetic experiments by placing optic fiber into the LH or nuclear combines respectively. And we found optoactivation of VTA VGlu2 fibers in the LH or nuclear combines consistently and strongly promoted waking from non-REM sleep in both two brain areas. So the above results suggest nuclear combines and LH are two main brain areas responsible for this VGlu2-induced wakefulness. And then I add one more slide here. So as for the VGAT neuron, we also did a chemogenetic whole brain C4 mapping by deactivating those VTA VGAT neurons because that promoted extended wakefulness. And we mapped the C4 expression in the whole brain. So we found C4 was elevated in the local VTA area, but not in the HM4DQ expression cells because this is the inhibitory jet receptor. And also we found C4 increased in the LH area. So we also stained those C4s in the LH with orexin, and we found the majority of those C4 cells are positive for orexin or hypercreatine. And because, as I showed, those VGAT neurons in the VTA are wake and REM active, so we speculate those VGAT neurons are not physiologically triggering non-REM sleep, but instead limiting wakefulness. So to explore this, first, we did channelrhodopsin-based circuitry mapping. We put channelrhodopsin into the VTA of VGAT mice, and we mapped the whole brain projections. We found dense fibers in the local VTA and LH area. So the especially dense fibers in the VTA indicate very strong local inhibition. So to test this, we infused GABA-A receptor antagonist GABAZine, simultaneously exciting those VGAT neurons in the VTA, and we found GABAZine reduced CNO-induced long-time non-REM sleep, but however, as blocking local GABA transmission did not completely block this CNO-induced non-REM sleep. So the projections to the LH might be involved in generating this extended non-REM sleep. So next, we placed optic fiber into the LH area and then inject channelrhodopsin into the VTA of VGAT mice. And then we found optostimulation VTA VGAT2 LH fibers strongly initiated non-REM sleep from waking. So the above results suggests the VTA VGAT neurons can limit wakefulness by local inhibiting dopamine and glutamate cells, as well as by projection to the LH. So in summary, so during natural wakefulness, the VTA dopamine and VGlu2 neurons generate wakefulness by exciting nuclear combines and orexin cells in the LH. On the other hand, the VGAT cells in the VTA provide an ongoing inhibitory tone onto those two types of neurons. And during natural non-REM sleep, all types of neurons are silent or fail less. So it has been shown that activation of dopamine cells produce extended wakefulness through nuclear combines circuits, and we found activation of VGlu2 and VGlu2 neurons promote extended wakefulness through nuclear combines and LH circuitries. And also, we found loss of weak neurons used in chronic lesioning the caspase, well, I didn't show here because I don't have enough time, induces extended wakefulness for about 4 months. And on the other hand, activation of GABA cells in the VTA produces non-REM extendation by inhibiting local VGlu2 or dopamine neurons, as well as inhibiting orexin cells in the LH area. So I would like to thank my supervisors, Bill Wiston and Nick Franks, and all the lab members in the lab, and all collaborators in China and the Crick Institute. And thanks for your listening.
Thank you very much, Xiao. We have time for a few questions.
Thank you, Xiao. It is a really impressive set of experiments. I have a general question. When you say that you induce wakefulness with your manipulations, is there a way to tell that this is normal wakefulness and not sleepwalking or some kind of state that resembles waking behaviorally, but, for example, animal cannot learn or is not fully there?
We didn't do any detailed observations, but we do find they were normal in their home cages. And we did locomotion experiments, and we didn't find they are hyperactive. So we think it's a kind of quiet wakefulness. And I think they behave normally.
Other questions? We are approaching the black hole zone that is lunch. You mentioned you've done the caspase experiments. Have you also tried to silence as well as ablate? And do you see differences if you've done it, or do you have any idea?
Yes, as I showed, we did HM4,
okay.
And we found inhibition of those big neurons produce extended wakefulness. It's like the caspase listening experiment.
So do you see that ablating a population or silencing the population has the same...
They produce the same phenotype.
The same phenotype, yeah. Okay. That's cool. More questions?
Yeah, great talk. I was wondering, what are the projections to these VTA neuronal populations?
It's quite different. So the dopamine has the same projection patterns as VGlu2 neurons. And we found, like dopamine, VGlu2 neurons project to, prefrontal cortex, lateral habenula, alpo, and lateral hypothalamus. But we found VGAT is slightly different. The projection intensively locates in the local VTA area and the lateral habenula and the LH area.
I couldn't resist, but what about the Niagara pass? Because they are exactly the same neurons, except that they are segregated between the complexa and the reticulata, and they are a very major gateway to the cortex.
Yes. So I think there's a paper online about the GABA cells in the Niagara that regulates sleep and wake. So I can remember what they found is the Niagara project differently. But activation of those GABA cells in the substantial Niagara can also produce non-REM sleep.
Yes, that's what I saw.
Yeah.
So it's similar.
Yeah, similar.
Okay. We have time perhaps with one more quick question. If not, we shall just thank Xiao again and move to the next speaker.